Hello students. So in my previous lecture I've already discussed what is bioelectric potential and how the action potential gets propagated from one cell to another, what is the propagation rate. So these things I have already discussed. Now in this lecture I'll be discussing how we actually going to measure the bioelectric potential. What is the need of measuring the bioelectric potential? First, let me tell you what is the need of measuring the bioelectric potential. So in the very introductory part of this bioelectric potential lecture, I told you that what is bioelectric potential. In the body we have various physiological systems, and these physiological systems are made up of certain specialized type of cells, and because of the electrochemical activity in these special type of cells, what happens, bioelectric action potentials or ionic voltages get developed across this cell membrane, and these ionic voltages are called bioelectric potentials. And these bioelectric potentials, they are actually the monitoring signals which actually display the functioning of that particular physiological system. So in order to know the functioning of the physiological system, whether the physiological system is functioning properly or not, so in order to know this, we need to measure this monitoring signal, that is bioelectric potential. So how to measure this bioelectric potential? So bioelectric potentials, they are basically ionic voltages. As I have already told you, what are they? They are the ionic voltages which get developed across the cell membrane. Okay. Now these ionic voltages, in order to display, in order to display these ionic voltages, we need to convert these ionic voltages into the electrical form. We need to convert these ionic voltages into the electrical form so that we can either amplify it, we can either process it, or we can display it for displaying purpose also. And for that we need a transducer. So we need a transducer which can convert this ionic voltages, or you can say the bioelectric potentials, into the electrical form. And which transducer can do it? That is electrodes. Electrodes, a pair of electrodes can actually convert the ionic voltages into the electrical form. So a pair of electrodes, it can actually convert the ionic voltages into the electrical form. So up to this point we have known that electrodes can convert the bioelectric potentials into the electrical form, and which can be displayed on the screen. Now, since as we have seen that individual cell produces action potential, individual cell produces action potential. So if we want to measure this action potential, or you can say the bioelectric potential, we need to insert electrodes into the individual cells, which in many cases will not be feasible. So what is the ultimate solution? So the more common form of measured biopotential, it is the combined effect of large number of action potentials, or you can say large number of bioelectric potentials as they appear at the surface of the body. So how these bioelectric potentials are actually measured? We use the electrodes on the surface of the body, and from the surface of the body we get the summation of action potentials, or the composite effect of action potentials of a large number of same type of cells. So suppose we want to measure the bioelectric potential of heart. So how it can be measured? We cannot insert an electrode into the individual cell of a heart. So what we will do, we will use electrodes at the surface of the body, which will pick up the bioelectric potentials, that is the combined effect of action potential of large number of heart cells. So this is how bioelectric potentials are measured. And when you actually going to display this bioelectric potentials on the screen, we will get a graphical representation of that bioelectric potential, and this graphical representation is actually identified, or the designation of the waveform, it actually ends with the suffix gram. For example, if I have actually displayed the bioelectric potential of heart, if I've displayed the bioelectric potential obtained from the heart on the screen, so the graphical representation so obtained, the graphical representation of the bioelectric potential of the heart will be called electrocardiogram. So let me show you how the electrocardiogram looks like. So this is the electrocardiogram. This is the electrocardiogram. This is the graphical. What is electrocardiogram? The graphical representation of bioelectric potential obtained from the heart muscle cells. So this is the combined, this bioelectric, this graphical representation of bioelectric potential, it is the combined or the composite effect of action potentials of a number of heart muscle cells. So in this way we have electromyogram, then we have other graphical representation of other bioelectric potentials of other specialized cells. So I will be discussing in those things in my next upcoming lectures. So till now you might have known that we have how the graphical representation of bioelectric potential for the heart is called electrocardiogram. Then we, in the similar way, we have electroencephalogram, and then we have electromyogram, etc. So first we will go through the heart part. Okay. So before actually discussing this graphical representation of the bioelectric potential, what are the various parts of this bioelectric potential actually mean, let me tell you how the heart functions. So I have already told you to go through the physiological systems in detail. Here I am just discussing in very brief so that you may know the details in order to understand this electrocardiogram graph. Okay, so let me discuss the functioning of the heart. We know the heart has 4 chambers, that is 2 atrium, right atrium and left atrium, and we have 2 ventricles, that is right ventricle and left ventricle. So the heart has 4 chambers, right atrium, right ventricle and left ventricle. Now the right atrium receives the impure blood from the various parts of the body through this superior vena cava. So through the superior vena cava, the right atrium receives the impure, or you can say deoxygenated blood from the various parts of the body. Then this right atrium, what it does, it pumps all the deoxygenated blood into the right ventricle. From the right ventricle, the deoxygenated blood goes to the lungs, where the blood gets oxygenated, and then the lungs send this oxygenated blood to the left atrium. From the left atrium, the oxygenated blood goes into the left ventricle, and from the left ventricle, the oxygenated blood gets supplied to the various parts of the body. So this is the functioning of the heart. So this is how heart functions, how oxygenation and deoxygenation of blood takes place inside the heart. Now let me discuss in more detail. So what is happening? The blood is getting pumped from atrium to ventricle. Now how the blood will get pumped from atrium to ventricle? The atrium had to contract. The atrium have to contract. Then all the blood will get to the ventricle part of the heart. So what is happening in this whole procedure? What is happening? The blood is flowing from these blood vessels to the atrium, and from atrium to the ventricles, and from ventricles to the either to the lungs or to the various parts of the body. And for this process to happen successfully, what is the requirement? The atrium has to contract. If the atrium contracts, then only the blood will go to the ventricle part. And from the ventricle part, if the blood has to go to the either to the lungs or to the various parts of the body, the ventricle again has to contract. So contraction of the heart muscles is necessary. And how this contraction of heart muscles can be achieved, that I'll discuss in my next lecture. So I'm ending this class here. In the next lecture, I'll be discussing how the contraction of atrium or ventricle is actually happening, and how this functioning of heart is achieved, or how the heart is actually functioning properly because of this contraction. The whole process that I will be discussing in my next lecture. So I am ending this class here. Thank you.